The next thing we want to talk about is some factors that can affect enzymatic speed. Number one thing that can affect enzyme speed is going to be substrate concentration. Enzyme activity increases with substrate concentration, which kind of makes sense. This is going to be due to more frequent collisions between the substrate molecules and the enzyme. Makes sense. If the whole point is to find somebody and you're sitting on a dating app, you're going to have more luck the more people that are using that dating app. If there are only 5 people out there on that app, the odds are that you're going to find one that you're compatible with are very low. It's going to take you a long time to make a match if there's not a whole lot of options on there. But if you increase the substrate concentration, you increase the number of people that are using that particular app, the odds that you're going to make a favorable bond go way up. So enzyme activity is going to increase with substrate concentration. The more people using it, the faster and better it's going to work. Now, temperature can also increase. Enzyme activity. The warmer temperatures cause more effective collisions between the enzyme and substrates. However, if the temperature increases too much, it can actually denature and destroy those enzymes. It can start to break those hydrogen bonds that are holding that enzyme together. Once we start breaking those hydrogen bonds, that protein is going to change its shape and it's not going to work it's not going to work very well anymore. pH. Most enzymes are optimized for a particular pH. So in the case of pepsin or trypsin, optimum protein digestion takes place at their respective pH optima. In other words, one of these enzymes is going to be used for digesting proteins inside of your stomach. For that, it's going to require an incredibly low pH. That would be our pepsin. Pepsin works best at a very low pH, very acidic environment. Trypsin, on the other hand, is going to be found elsewhere in the body. It's going to be found in the small intestine. And in that case, we actually don't have a very acidic environment. So we don't want an enzyme that's optimized for a low pH. So trypsin is going to be optimized for a higher pH. And once it becomes too acidic or even too basic, enzyme activity is going to drop off. And so those are sort of things that can regulate enzyme activity just on a physical level. But there are other ways that the cells can help regulate enzyme activity. Cells can actually regulate the presence or the absence of an enzyme. We can actually start producing enzymes, or we can stop producing enzymes. This is going to be done through gene regulation. So enzymes can regulate the presence or absence of an enzyme. Enzymes can regulate the concentration of an enzyme. So we can say, hey, we need this enzyme, we don't need that enzyme, we need more of this enzyme, we need less of that enzyme. So we can actually dial it up or turn it down to help drive those reactions forward. We can also activate or deactivate some enzymes. A lot of times this is going to be done through enzyme cofactors. Cofactors are going to be molecules that are required to activate enzymes. Great example of this would be FAD and NAD plus and NADPH. Or an NAD+. These are going to be cofactors. The first two are going to be involved in cellular respiration, which we will see in Chapter 8, and the third is involved in photosynthesis, which we will see in Chapter 7. Coenzymes are going to be non-protein organic molecules that are going to play an important role in activating or deactivating those enzymes. Great example of these would be vitamins. Vitamins are small organic compounds that are required in the diet for the synthesis of coenzymes. This is why vitamins are so very, very important. You need these. You can't produce them yourself. You need them in order to produce the coenzymes that are going to regulate enzyme activity. So we're going to talk about how we can regulate enzyme activity, and the easiest way to regulate enzyme activity. If we want to increase enzyme activity, we're going to just make more enzymes. If we want to decrease enzyme activity, we can either stop making enzymes or we can actually inhibit the enzymes we currently have. We can do this through a process known as reversible enzyme inhibition. In this case, when we're doing reversible enzyme inhibition, we are going to, a substance known as an inhibitor is going to bind to an enzyme and decrease its activity. It's just going to slow that enzyme down just a little bit. We don't want to shut down the enzyme. We don't want to remove the enzyme. We just want to slow the reaction down a little bit with an enzyme inhibitor. We can do this one of two ways. We can either do competitive inhibition. Competitive inhibition is when the substrate and the inhibitor are both able to bind to the active site and they're going to compete with one another. So the product is only going to form when the substrate binds to the active site. And again, this is a really great talk to have with my chalkboard, but since I do not have that, I'm going to just recommend that you sit in for our discussion group when we talk about competitive and non-competitive inhibition. But I'm also going to recommend that you find some YouTube tutorial videos that are going to cover these topics and bring them to our discussion meetings as well. But competitive inhibition is where the product is only going to form when the substrate binds to the active site. And we're going to just have a bunch of other inhibitor molecules that are going to slow it down and compete with one another. I don't know if any of you have ever done this, but when I was little, I went to a friend's house, and they were feeding their cat, and inside of their cat's food bowl was a whole bunch of marbles. And I was like, why are there marbles in the cat's food bowl? And they said, well, our cat is kind of a glutton, and she eats so fast that she's going to make herself really, really sick. So we have to slow her down by putting marbles in her food bowl. So there's now food and there's marbles in her food bowl, and she can only eat when she comes across a food particle. So she'll, eat a food particle, and then she has to dig through a bunch of marbles to find another food particle. Then she'll dig through a bunch of marbles to find another food particle. It just slows down her eating, so she doesn't get sick. This is the way competitive inhibitors work. They're going to bind to the same site. They're still going, towards the mouth. It's just when a marble goes into the cat's mouth, the cat's not going to eat it. It's going to spit it back out. It's only going to eat it if it comes across a food particle. So it's just going to slow it down a little bit. They're both there. They're both going to bind, with the cat's face. I'm sorry, this analogy is starting to get a little bit weird. The whole point, though, is that we are competing for the same spot. We're only going to make our product if the substrate binds in there. If the inhibitor binds in there, nothing's going to happen. But it's not going to stay in there. It's going to move in. It's going to bounce out. We're going to bind to the active site. We're going to unbind from the active site. We're constantly moving in and out and in and out. When the competitive inhibitor is bound to the active site, nothing happens. Then when it hops out and the substrate binds to the active site, we're going to produce our product. So we're just kind of slowing the process down. Non-competitive inhibition is a little bit different. The inhibitor does not bind to the active site. It binds to another location called the allosteric site. When we bind to that allosteric site, the change in shape initiated by the inhibitor binding to the allosteric site changes the shape of the active site, making it unable to bind to the substrate. If you look at our allosteric site, what happens is, here's an example of our non-competitive inhibition. Our substrate A is going to bind to enzyme 1. And when it binds to enzyme 1, it's going to convert to product B. Product B will bind to enzyme 2, convert to product C. Enzyme 3 will help it convert to product D. Product D will convert to convert to product E. So on and so forth. So here's what that metabolic pathway. Now here's the interesting thing. A lot of times we will use the end product F. Once that end product F is produced, it's going to bind to the allosteric site of enzyme 1. And when it binds to that allosteric site, it's going to change the shape. It's going to change the shape of the active site. And once we've changed that shape, see where, we have this sort of harsh, triangle shape. Enzyme A or substrate A cannot bind. So substrate A cannot bind if the end product or F is bound to the allosteric site. Now this is one way that we help regulate metabolic processes in the body, because the whole point of substrate A binding to enzyme 1 was so we could make so we could make B, so we could make C, so we could make D, so we could make E, so we could make F. Once we have F, do we need A to still be going through this process? No. So once we have plenty of F, we're going to bind to the allosteric site, we're going to start turning A away. Now, once we use up our end product, once our end product gets shipped out of the cell or it moves on to wherever it needs to go, it's going to bounce out of the allosteric site, and the active site of enzyme 1 is now going to suddenly open up and be available for substrate A again, and we can open up this process. Once too much F builds up, this process is going to shut down. Starting right back here at the beginning, we're going to shut down. We'll stop converting A to B. Then once we use up F, it'll go away, and production can begin again. It works very much like your thermostat. Your thermostat waits until the temperature gets too high, and then it shuts off, and then it waits till the temperature gets too low, then it kicks back on, and then once the temperature gets too high, it shuts off, and this is how we can regulate the temperature inside of a house. So this is non-competitive inhibition. Again, this is a really great opportunity for you to send me some extra credit videos in our discussion groups. We'll probably watch a couple of videos together. There's some really great tutorial videos out there. I would love for you guys to see them. They're very interactive, and I would love for you guys to have a chance to look at those and play around with those. So I'm going to set that aside for now, and we will talk about that in our discussion groups. All right, to summarize this chapter, we talked about cells and the flow of energy. We talked about how energy is the ability to do work or bring about a change. Kinetic energy is the energy of motion. Potential energy is stored energy. The laws of thermodynamics, we talked about how energy cannot be created or destroyed, only transferred or transformed from one form to another. One form of usable energy cannot be completely converted into another form of usable energy. That is our second law of thermodynamics. Entropy is always increasing. We're going to be losing usable energy in every single transformation. Metabolic reactions and energy transformations. Metabolism is going to be all the chemical reactions occurring in a cell. We talked about exergonic reactions and endergonic reactions. Exergonic reactions occur spontaneously. They're going to release energy. Endergonic reactions, on the other hand, are non-spontaneous. They're going to require an input of energy. ATP is going to be our energy currency of the cell. We went over its shape, structure, and function, so please make sure you take time to review that as well. Then we talked about metabolic pathways and enzymes. We talked about how a metabolic pathway is a series of reactions that proceeds in an orderly step-by-step manner. Enzymes are going to speed up those reactions by lowering the activation energy. And enzyme activity is going to be influenced by things like temperature, pH, the presence of cofactors, and or inhibitors. Make sure when you're going over this chapter that you review inhibition, you review competitive and non-competitive inhibition, and that you're familiar with the term allosteric. And I think that's it. That's all we have for this chapter. Let me know if you have any questions, comments, or concerns.